It's my pleasure to introduce you to Dr. Alessio Fasano. He's the Chief of Division of Pediatric Gastroenterology and Nutrition and Vice Chair of Research at Mass General for Children. He's also Director of the Mucosal Immunology and Biology Research Center and the Center for Celiac Research and Treatment. He's also serves as Professor of Pediatrics at Harvard Medical School and is a Professor of Nutrition at Harvard T.H School of Public Health. Oh my gosh, we are so honored to have you here. In 2000, his group discovered zonulin. This is the protein you've probably heard about, associated with gut dysbiosis. It regulates intestinal tight junctions. He'll talk about that. His basic research focuses on bacterial pathogenesis, the gut microbiome composition, and the function of the health and disease, as they all relate to each other in respect to gut permeability, mucosal biology, and all the way through the immune system interacting with even the SARS virus, the COVID virus. He's also the author of 2 books, Gluten Freedom, and he's co-authored another book called The Microbiome and Our Oral Health. I have a copy of it here. I know a couple of you brought copies for him to sign. Fantastic book. It gives you a really broad scope and deep perspective on the role of microbiome in across the health spectrum. A fascinating reading, easy to understand. So I really encourage you to explore that for your reading this summer. And we're really honored to have him at SCDROCKS to provide our keynote presentation on how nutrition can impact the composition of the microbiome and its relationship with permeability and the immune response. Dr. Fasano.
Thank you, Ali, for the kind introduction. And again, I'm thrilled to be back in Seattle, also because it's not raining. In general, when I come here, it rains. But anyhow, and again, just a disclaimer. Whatever we make with this book, we're not going to make any money. It goes back to science, so we use that for support the research. When Jeffrey and Ali invited me to this meeting, they made a strange request. They said, you know what, would you mind to make this a real story rather than, the typical presentation? And I knew immediately what they were talking about because, again, for people that are in science, they know well that what you read in this book or, in a paper or in a magazine or on the YouTube and so on and so forth is the sanitized version of what is the real world of science. I keep saying to my students, science is a constellation of failures with very few successes. And if you will be here to listen to the failure, will be extremely boring. So you hear only the successes. And, when we present this paper, it seems like they were so smart that we thought about this and again, make a kind of discovery. But, serendipity plays a great role, in making discoveries, and sometimes they can become transformational. So this is going to be a story of serendipity. They started, a while ago. You heard that I am a pediatric gastroenterologist by training, and I've been always interested in diarrheal diseases. And again, seems to be not glamorous, but until the recent past, we lost 5 million kids every year to diarrheal diseases like the one against cholera, for example. And when I moved to the United States first, that was my job, to develop a vaccine against cholera. And again, I was young and very naive, and I was absolutely sure that I would take care of this. And indeed, after a couple of years working extremely hard, I developed this vaccine, and it worked beautifully, absolutely beautifully in animal models. It did exactly what I was supposed to do. And at that point, you need to give it a try in human beings. So we are now, let's see, late 80s. What do you think the volunteers that will take a vaccine against cholera will come from? Any idea? Who will be the people that will be so nuts to say, yeah, I'd do it for money, of course? Who? students, medical students, exactly that. And what was the premise was, this is a great deal because you have 3 chances here. One, you got the placebo, so water, so you take the money for nothing. The other one, you got the vaccine that I can reassure you is not going to hurt you, so you get the money for free. There is one chance that you got the real deal. You poop in your pants for several days, but we take care of it. Don't worry about it. So you're going to survive. Well, I started to do this. At that time, was very cool, engineer to cut, the genes that make this cholera to give you this purging diarrhea and so on and so forth. Got this vaccine and gave it to the students, and this was the outcome. So it didn't work. So it was not the purging 30 liters of diarrhea that the kids that got the real deal end up to experience. 30 liters that they have to go, and get care for, but was still too much diarrhea that was not acceptable. So this is one of our students. You see the face because he said that was okay. He has in his hands what we call the hat. So when you do these studies, you need to measure how much poop they get there. And on the bottles there is the IV fluids that we have to give him to make sure that he didn't get dehydrated because of this vaccine. So it didn't work. Literally, 2 years of science flushed in the toilets. That's pretty much what it was. So what you do in a situation like this? Well, people react in a different way. The way that I reacted, I got drunk. I didn't show up in the lab for a few days. People start to be worried about me because it's suicidal, whatever happened. But eventually then you go back to the drawing board and say, why? Why did that happen? How come that, the vaccine was devoid of the genes that make this cholera toxin that makes the people to have diarrhea? Why there is still diarrhea? And then how you eventually discover something else, a new toxin with a different mechanism of action that was by making the intestine leak, so in other words, increase the permeability, that eventually led to this discovery that we published. And of course, when you do this kind of study, you ask yourself, okay, how this toxin works? What is the mechanism? And then you realize the mechanism is so complex that you wonder why we human beings have all this machinery of complexity of signaling to make the intestine leak, just to be the target of a toxin, of a bug that make us sick. Gotta be something else there. Gotta be a mechanism that we use for different purposes that cholera learned during evolution and exploited for its own purpose, i.e. to increase, the fluid in the gut. And that led to the discovery of this molecule. In 2000, we discovered this molecule, zonulin, as the counterpart that modulate this permeability of the gut that was targeted by cholera. This is serendipity at work, And now, fast forward now, this molecule has been linked to a long list of chronic inflammatory diseases, from neurodevelopmental problems to autoimmune diseases, to metabolic disorders, to cancer, to aging, to food allergies, infections, and so on and so forth. We're not going to be able to review all this, but now this has become a biomarker of a variety of conditions that seems to be all disconnected to each other, but it's a common denominator right there, inflammation. These are all conditions that have a connotation of inflammation. And, I want to bring this now in the context of what you guys care for, a close interaction of the food, the gut, the antigen trafficking, therefore the permeability, but most importantly, something that is very dear to me, this communication between the gut and the brain. It can lead to some feeding disorders, and that we're not going to be able to look at all this. But putting this in the context of our lifestyle and why you guys are sitting here. These chronic inflammatory diseases had a kind of interesting journey. you know, just to remind you, the human being that has a 2 million history has got sick and died mainly by one and only one reason: infections. That's the reason why we got sick and died. Until the very recent past, the last few seconds of our evolution, and that happened mainly in the Western Hemisphere. When we had the understanding and the grasping why we got sick and died of these infections, we start to really developing science like, you know, infectious diseases, bacteriology, and so on and so forth, that led us to deploy, and mostly only the Western Hemisphere, tools and interventions to mitigate the impact of infection on our health. The discovery and use of antibiotics, the development of vaccines, hygiene of the waters, and so on and so forth. And therefore, ever since we've been implementing this measure to ameliorate infections, we have seen a dramatic drop, as you see on the left side here, of infectious diseases like rheumatic fever, Hep A, TB, mumps, measles, and so on and so forth. Dropped dramatically. However, during exactly the same period, we've seen at the same latitude a dramatic increase, or in the opposite direction, on non-infective chronic inflammatory diseases like Crohn's disease, MS, type 1 diabetes, and asthma. Bottom line, the lesson that we learned from here was the following. Now we don't die fast of infectious disease as we used to. We die slowly of chronic inflammatory diseases if we embrace a Western lifestyle. But there was another lesson that for me was even more important. There was an eye-opener situation for me because I was not aware until, you know, this that was called the hygiene hypothesis, and then the completion of the Human Genome Project put on a table. At that time, again, I was still young, no white hair yet. The premise was one gene, one product, meaning one protein, one disease. That's the reason why we embraced in the Human Microbiome Project. Meaning, if I have the genes for Crohn's disease or for multiple sclerosis, it's a matter of time. I'm going to come down with that. And there is nothing that I can do about it. That was the premise at that time. The results of the Human Genome Project was that we discovered that, genetically speaking, we are extremely rudimental. We are made of only 25,000 genes. That's all. If you are a fisherman or fisherwoman and you go fishing, that worm that you use to go fishing, 75,000 genes, 3 times more than we do. A plant like wheat, 150,000 genes. How come we're on the top of the scale of evolution with only 25,000 genes? That really bothers me. But that's an object in another discussion. But honestly, that premise, one gene, one protein, one disease, went out of the window. And the lesson that we learned was genetics is extremely important, but it's not that simple, a one-to-one solution. It's much more complicated. But the other thing that we put on the table as a premise, and we were highly criticized at that time, was, you know what? If I'm genetically predisposed for Crohn's or diabetes or colon cancer, that's not destiny. If I do or do not develop that disease, depending how I play my genetic cards, i.e. my lifestyle. Because that's the only way to explain that. And that brought us to this paradigm that, again, was put forward almost 2 decades ago and was, again, highly criticized. You know, the news have very few friends in general. But, you know, now I think that nobody would dispute that. Genetics and exposure to environmental triggers remains absolutely essential, necessary, but not sufficient. There are at least other 3 elements to have the perfect storm that leads to disease, any given disease. One is these 2 worlds are normally segregated. You know, the genes are express stuff in our body. These environmental triggers are outside our body, and we have barriers that defend us for this uncontrolled, encounters of these elements that come in our body. The most obvious that we see is the skin, but there are many other barriers. The largest of all is the gut, because if you take the gut of an adult and you spread on a floor, it will cover half of this room, so 200 square meters. So it's pretty large. And if you have a loss of the barrier of the gut, what we call increased gut permeability, or quote-unquote leaky gut, terms that I don't like very much. But anyhow, these two worlds now, they are not segregated anymore. So these enemies can come in and can instigate what? Inflammation. And therefore, the fourth element is an immune system that becomes hyper-belligerent when under attack. And last and not least, because you heard this again, and you will see how important this is, the microbiome. It really dictates our destiny. Now, these elements are being put out there, artificially divided from each other. But you will see in a moment, they are highly, highly interconnected, particularly the last three. If you have an increased gut permeability, you have an increased antigen trafficking, so junk that comes from the environment in our body, and that will make the immune system to become hyper-belligerent. And vice versa, an immune system that fights all the time will increase your gut permeability. Same story, gut permeability will increase your dysbiosis and vice versa. You got the point. But the lesson that we learned that now makes sense, what was inconceivable, i.e. how we can be so complex with only 25,000 genes, is the pressure that the microbiome put on our genes, what we call epigenetics. In other words, it is the environment that through the microbiome would decide if, when, how, and when these genes will be turned on and off, starting the march from genetic predisposition to clinical outcome. So having those genes is necessary, but doesn't mean that I would develop this disease. It really truly depends on how our microbiome would touch those genes and start that march. These are complex stuff, so I got to stop for a second here. Am I talking too technical? Did I lose some of you guys here? Are we sure? Because otherwise I start all over again. No. Okay. All right, so what I just told you is summarized in this cartoon. It took a while to put this together, but follow me for a second. This is the march from health to the disease of all of us. If you start left, that's health. Number one, the barrier works, the segregation of the enemies from that can create inflammation is maintained, they stay out. The mucosa, in this case, the intestine, because again, this is an example, but take the lung, take the gastrointestinal tract, even the skin is the same story here. That barrier is defending me, and I reach what we call a state of normal physiological energy, homeostasis. I'm healthy. The problem arises when we go from point 1 to point 2. That barrier doesn't work anymore, particularly the spacing between cells that are regulated by structure that you heard from Ali, they're called technically tight junctions. Consider them as sort of doors. They're always closed, but because of doors, they can be opened when needed. And if they are not open for too long, you open a shortcut and start to have not a controlled antigen trafficking as in number one, but a flow of this stuff coming in. And if they do, of course, what happened? The immune system sees the enemy coming, and the immune system is being programmed to fight enemies. And when you fight, there is collateral damage, there is inflammation. This inflammation is mediated by some chemical substances that we call chemokine or cytokines that per se increase gut permeability, and you got in this vicious circle. Antigen trafficking, inflammation, further permeability, more antigen trafficking until you break tolerance and you develop a disease. Which disease? Depends who you are genetically speaking. Again, that's the reason why genes are important. If you are predisposed, let's say, for functional disorder, you develop IBS. Type 1 immune response, you develop cancer. Type 2 food allergies, type Th17, autoimmunity, and so on and so forth. So that's the essence of the story. And our contribution to understand the heart and soul of why and how you start the march from genetic predisposition to clinical outcome has been indeed the moving from step 1 to step 2 that is mediated by zonulin. That is one of the many, there got to be others we didn't discover yet, molecules that can eventually break that compartmentalization that keep the enemies out and will start that march. When we do this kind of stuff, we in general use animals to prove mechanistically that indeed that's the case. And again, I don't want to spend too much time, but I want to just show this just as an example of how this triangulation, gut permeability, immune system, and microbiota really works. So these are mice that they produce a lot of zonulin. They've been genetically manipulated to produce a lot of zonulin, and therefore their intestinal leaks. We have a way to measure that, even if there is no damage in the intestine. So this is a functional problem and not a structural problem. And when you look at the microbiota of these animals, again, they are in dysbiosis. If you compare to normal animals, the one in green, the zonulin transgenic mice, so the ones that they generate a lot of zonulin, they have a different microbiota, completely segregated, that you see in blue, no matter if they are males or females. And when you ask why they are different, what are the signature that make the difference in this microbiota, is that the zonulin transgenic mouse lost protective elements in the microbiota, like Akkermansia, and has an excessive representation of pro-inflammatory microorganisms like Rickenella. So in other words, this animal is ready to generate inflammation because of this dysbiosis and this antigen trafficking. Not only that, but if you look at the immune system, again, don't get scared because I don't understand this either. So the immune system is also different. So in other words, these animals, compared to the normal mice, they have less immune cells that puts brake on inflammation. So the ones they have to just make sure that we don't do too much inflammation. And they have an oral representation of cells that they generate inflammation all the time. So see the perfect storm there. Now, these animals, if you don't do anything to them, they are normal. They grow well. They don't have any major problem. Interestingly enough, they're behaviorally different from the normal mice. They have strange behavior. They have an ADHD-like behavior. But in terms of growth, reproduction, and so on and so forth, they're indistinguishable unless you touch them with a stimuli that eventually create a problem. In this case, a stimulus that induces inflammation. That is a chemical that we give them. And when you give this chemical, these animals, 70% of them die compared to 0% of the wild type. So if they are under stress because of the stimuli, they have that problem. And they are completely rescued if you use an anti-zonulin molecule that now is in clinical trials for many conditions, that is the larazotide. It's a synthetic peptide that blocks the pathway, and therefore these animals from 70% went back to 0 mortality. Just to prove that this is all zonulin mediated. So why have I did this excursus? Just to let you know that, again, that triangulation, gut permeability, hyperbiligen immune system, dysbiosis, it's going to be fundamental, together with the other two elements, to really move from health to disease. Now, when I was, again, a young researcher in Baltimore doing this cholera vaccine, the concept at that time was all microorganisms are, pathogens, and we need to fight them because they kill us. Remember, that's the reason why we start this journey of, against infectious diseases. And I was doing this by developing a cholera vaccine. Well, that was a myopic view because we learned over time that the pathogens are the tip of the iceberg of a much more complex ecosystem, made mainly almost 95% of microorganisms, they are friendly. They really want to live in a friendly relationship with us. These are the ones that we're unaware because we could not cultivate them on a petri dish. That's we typically are able to do with pathogens because they have a biological lifestyle that make very, very difficult to visualize them under the microscope. Why we got in this, symbiotic relationship? There are a lot. Some people believe there are even more microorganisms than our own cells. So if I want to be very objective, I'm not looking at, 50, 60 people here, but I've been looking at the zillions of bacteria and viruses and parasites looking at me right now and wondering where he's going with this discussion. And complexly, they make more than 100 times the genes that we do. Their advantage to be in a symbiotic relationship with us is obvious. They have hospitality. They got food from us. What are we going to get from this? Why we got to do this? Well, we got a lot. In exchange of that hospitality, we got extra calories from food that we cannot digest, for example, fibers. And it's probably the most important function they dictate if we're going to stay healthy or not, they regulate our metabolism in functions that can really dictate if we stay healthy or not. They produce essential vitamins that we cannot have in a sufficient amount through the diet. As I told you, regulate the immune system, you saw that. And most importantly, they protect against the bad guys. After all, penicillin is nothing else than a molecule produced by a bacterium to kill another bacterium. But all this, all the mutual benefits, is important, but will materialize only and only if we are in a balanced, friendly, symbiotic relationship with our microbiome. And it's very personalized. Meaning, as we are all genetically different, even monogenetic twins, monozygotic twins are different genetically because epigenetics makes the difference. So is our microbiome. So there is no such a thing as a normal microbiome. What is normal is, so we are all genetically different. We all have a different microbiome. But the outcome of this interaction has to be, if we want to stay healthy, within a certain range. We all have to have a certain blood pressure, certain level of glucose in our blood, and all this is achieved by this interaction with the microbiome. You're not going to see a squat here, and that's okay. But the point that I want to make with this part of the slides is no matter what kind of environmental trigger, one of the 5 pillars to develop disease you want to consider, prenatal, what has been the lifestyle of my mother? Was she smoking or drinking? Was she living in an urban or rural area? Was she exposed to pollution? Perinatal, am I be born by C-section or vaginal delivery? Am I be born earlier so that I had to go in the intensive care unit? What kind of food I've been given? Breastfeeding or bottle feeding? Or postnatal? How many infections I got? What kind of lifestyle I've been doing? Am I under stress? All this have been shown indisputably to affect the composition and function of the microbiome. And that can affect the permeability and start that walk from genetic predisposition all the way down to the clinical outcome. Why we see these epidemics of chronic inflammatory diseases in the Western Hemisphere? Not the Agena hypothesis. That was the simple way to understand that. It is more that we derail from the plane of evolution. If we go through the plan of evolution, we will be at the left side of this other part of the slide. We will be born by vaginal delivery. This was the way that we're supposed to be born. We will be fed real food and not junk. No infections, because, again, 2 million years ago, life expectancy was 14 years. Either you die of infection, so you don't survive of that, or a dinosaur will eat you. There is no time to develop cardiovascular diseases. Of course, there were no antibiotics, and that make the balanced microbiome to be at the right place. Now again, the immune system was invented by evolution to fight one enemy. Again, infections. There was no cancer at that time. Meaning that particularly in this crucial period of time that you will see in a moment, the first 1,000 days of life from conception to 2 years of age, that's how we're going to dictate how we will play our genetic cards, believe it or not. Inflammation is a good thing if controlled. An immune system, that's what it does. Generate inflammation when they're under attack. Why? Because generates a very unfriendly environment for microbes to grow. It's too hot. There are chemicals like cytokine and chemokine that will kill me. Cells will come in, like immune cells, that will eat me. You pay a price, because the tissue that's inflamed will die, but the whole organism will survive. So if inflammation is used properly, it will be turned on when we are under attack, and will be turned off when the enemy is being defeated. That's not happening anymore if you embrace a Western lifestyle. And you are on the right side of this session. A lot of people, they do C-section that, by the way, is totally welcome if it's medically indicated, but not to organize the holidays of our OBGYN. That's not appropriate. You will see in a moment, but again, I know that I'm preaching the choir here. Inappropriate nutrition definitely will impact negatively in what is going to be the way that you play your genetic card. Multiple infection, a lot of antibiotics. You put your microbiome in dysbiosis, and now that imbalanced microbiome will program the immune system in a wrong way. Will put the bar to unleash inflammation very low, even when it's not really needed. And this continuous inflammation on a genetic background, sooner or later, will lead to breakout tolerance and start that march that I show you from genetic predisposition to clinical outcome. That's our understanding why we see this frequency of chronic inflammatory diseases. Even biology would tell us it is instrumental to maintain the microbiome imbalance. We nutritionists for many years have been battered and have this headache to understand the function of some of the elements that compose the breast milk, particularly the sugars. These human mono-oligosaccharides are very rich, are the most abundant sugars in the breast milk. We didn't understand what they were doing there, because metabolically, the babies cannot use those sugars for nutrition. What they're doing there, they nourish the microbiome to make sure that engraftment will be compatible with the baby. So they are phenomenal prebiotics to make sure that the good guys, the probiotic component, the microbiome, will have a chance to engraft. When this engraftment starts, we thought it was a birth during the passage of a vaginal canal if this baby is lucky to be born by vaginal delivery. Well, actually, it seems that this starts even before delivery. And that's the reason why mom lifestyle seems to be so fundamental. So there is even now evidence that during conception, there is a transfer of microbiome from the father to the baby. It's questionable, but there is now evidence growing in that sense. And there is now evidence of the blood microbiome, there is an evidence of placenta microbiome, and so on and so forth. But bottom line, all this to say that the mom lifestyle, particularly in the last trimester, can affect what composition that the microbiome will be transferred to the baby at birth. And therefore, it can be impactful. What is the destiny of this child? We're talking on the table, very early onset, IBD, 6-8 months of age. Genetics has a huge elements there, but epigenetic as well. Because again, if mom was exposed to pollutant or whatever, or antibiotics, she can transfer the wrong microorganisms. We've been studying this engraftment, what happened in the microbiota when the baby is born. It's completely chaotic. It makes no sense. So at the beginning, there are some microorganisms that engraft, then they leave. There are others that come in their place. Then baby food is introduced, and you have another shift. But everything quiets down around the 2 or 3 years of age when the microbiome reaches the maturity of the adult microbiome. Chaos? Nope, not at all. Dating. We are searching for each other. We are choosing each other. We're trying to find that sweet spot in which I, as a microorganism, like you as my host, because you can provide what I need from you. And I, as a host, like you to stay because you're compatible with my genetic makeup to keep me healthy. Meaning, anything that affects that searching each other can have tremendous consequences over time. Anything that will affect that searching each other. Again, external environmental factors may eventually put me in disadvantage to be in he agrees or she agrees, in disadvantage in a trajectory how I will play my genetic card, okay? My second part, how much time I have yet? Is I want to give you some examples of how all what we discussed that was the basic science to give a sense of what is the clinical outcome has been studied so far. And I choose an example of autoimmunity, an example of obesity, aging, because I'm extremely interested in that for myself. So just to give you an example of what we stand right now and what the future lies in terms of how we may make use of this information to play as better as possible our genetic cards. You heard that I directed the Center for Celiac Research, and that's the reason why I choose, as an example, celiac disease as a paradigm of, an autoimmune disease and its impact with this triangulation between permeability, microbiome, and immune system. We had this very complex study that is, the acronym is CDGEM. This is a prospective cohort of babies at risk for celiac disease because somebody in the family has it, so they have a higher risk to develop it, that we follow from birth until eventually they develop the disease. Just because, again, that's the only way that we can understand them march. So we monitor everything that you can imagine, mostly nutrition. So we have a very detailed nutritional diary of these kids. But of course, we measure, their microbiome, their genomics, the metabolomic profile, and so on and so forth. But again, just to understand why some kids, they take the wrong turn and how do they stay healthy. And same with other conditions that we saw, same story in celiac disease. Yes, genes and environment, that by the way is known here. We know that gluten is the culprit for celiac disease. We don't know what is the culprit for all the other autoimmune diseases, but we know that gluten is the culprit. So it's traceable. We know when it comes into the picture in these kids. They are necessary but not sufficient because some people, they are genetically predisposed. They eat gluten for 30, 40, 70 years and they stay healthy. And then eventually they come down with a condition. How? Epigenetics. Those genes that eventually start the march were not touched until later age. And other folks eventually will start much earlier because these genes are touched earlier. So what we found here, again, I don't want to go too much in the technicality of the matter, but definitely, as I show for the mice, these folks, they have a different microbiome signature if they develop celiac disease compared to the one that they do not. But what is intriguing is that that signature is present months, actually years, before that they come down with the disease. Some of these microorganisms, for example, these 5 that you see here, they're all protective microorganisms, are lost a year and a half before that these kids that develop celiac disease compare the same kind of predisposition of other kids that would not. So in other words, just these 5 will give us roughly 90% confidence that if they are not there, these kids will develop celiac disease within a year and a half. This is mind-blowing if it's going to be confirmed. And this is been done by artificial intelligence. So in other words, take all this data that we have and do modeling. So if this turns to be correct, and by the way, this is not specific strains of probiotics that have been linked just to celiac disease. There are some that have been linked to the risk to develop IBD or other chronic inflammatory disease and so on and so forth. But all this to say, imagine if this will be confirmed. And in the future, we have the capability to look at the microbiome of kids at risk of celiac disease. We see this, we provide these strains, and we intercept an autoimmunity. This is going to be a game changer if this will be confirmed. Obesity. So that's something else that really bothers me a lot. Of all the things that I'm a pediatrician, that's the reason why I'm very concerned about this. Of all the things that will eventually affect us big time in terms of morbidity and mortality, this is it. I mean, this is a map compared 1980s to 2015 of the obesity in boys and girls. The darker is the area, the more impactful is the obesity. See what kind of change we had in such a short period of time. Only 35 years. And you know, the CDC just, and UNICEF just came out with the last numbers, and you know, it's read everywhere. It is really, really concerning. This generation of kids that have that kind of problem will have a life expectancy lower than their mothers and fathers. This is the first time in human history that happened, that the next generation will have less life expectancy than the previous one. The reason why this bothers me the most is because this, as you will see in a moment, is not just lifestyle, it's also social. This is the map in the United States where we see this most impactful in the South. Why? Socioeconomically in disadvantage. These people, they do not have the means to feed their kids the right way. And indeed, there are biological, psychological, psychosocial, and social aspects of the story. I was mentioning, you know, before on the table, if I am a mother, single mother of 3, and I have $10 a day to feed my kids, what do you think that I'm going to feed? Fruits and vegetables? Can't. I need to fill the belly of these kids. So the obesity was the pathology of the rich in the 18th century, is the pathology of the poor in the 21st century. So we really need to pay attention to this as a social discriminating, you know, feature. And just consider what comes with that. The bullying of the kids that are obese, the pathology that as a pediatrician I've never seen before. We see cirrhosis now in pediatrics that was unheard of. We see cardiovascular disease in pediatrics that never occurred to me that could happen. That's the reason why the life expectancy is going to become low. And, of course, the microbiome seems to have a role. So if you're born by a lean mother, chance exists that you will receive microbes that will keep you lean, so to speak. But if you are born by a obese mother, she will transmit microorganisms that are capable to scavenge extra calories from the same amount of food that you ingest, and therefore we put these kids at risk. Of course, you say, well, of course, how you can make this point, given that, you know, there is the social aspect, there is also the genetic and so on and so forth. Well, because, you know, these humanized animal experiments, if you take the stools of an obese, you know, mother, you transfect a germ-free mouse, that mouse will become obese, even if the mouse will eat the same amount of food of the ones that would not be transfected. So all this to say, maternal health, birth mode, antibiotics, breast milk with the oligosaccharides that I was telling you, and so on and so forth, they all impinge on what's going to be the engraftment, and an altered gut microbiota will increase the risk of child health. And why? Same story, the mechanism that I told you before. Because if you have dysbiosis, you have a leaky gut, the Western dietary patterns would generate, you know, inflammation in your adipose tissue, and so on and so forth. Aging. That's another interesting area here. So now people, they talk about inflammation because even aging is a process of inflammation. What we didn't know, and this is not a mind-blowing, well, we had a sense. There are people that age very graciously and other people that crash. You all are looking very, you know, gentle aging here. So how come? It's all driven by the inflammatory process that is determined by genetics for sure, but also by lifestyle. So Why? Because it looks like that there's potential for physical, mental, and social growth of development, even at very late age. And there's been even more done in the science. The possibility of the fountain of youth existence has been proved because you have now evidence of rejuvenation, extending the lifespan of model animals, giving hope of what we call a negligible senescence. We got to die at some point, but without disease, being independent and active, that's the dream of all of us. Not to live eternity, that would be a curse, but to live healthy until it's time for our demise. So there are a lot of theories of aging. The three that now seems to take more of steam are the immune system theory of aging, inflammation and aging, and intestinal permeability of aging. And by the way, same kind of triangulation here as well. So this is a paper that came out a while ago reiterating that concept. Do you remember the zonal transgenic mouse? Same story here. Age-associated dysbiosis promotes increased gut permeability, systemic inflammation, and some of this immune cells dysfunction that leads to the inflammatory process of inflammation. And if you look at, the microbiome per individual that they, or during their lifespan, they differ in some of their composition. So aging comes with change in the microbiome as well. This is the one that I really find extremely interesting. Fruit fly. You say, who cares about the fruit fly that we're far from them? Well, let's learn here. What is predictor of the death of a fruit fly is not its chronological age. It's when the intestines start to leak. That is setting their lifespan. So in other words, the inflammation of, senescence, aging, is due to the fact that we all, from birth to death, have to balance between destruction due to inflammation and capability to repair. But when we really got to the senescence, the elements of destruction, they start to be more and more impactful, and our capability to regenerate, is less effective until we reach the point in which there is no way to compensate, and that's going to be all destruction that will accelerate and not enough correction, and we die. They found a way to stop the leakness of the gut of this fruit fly, and their life expectancy doubled. It's like to say, if we stop the leaking in our gut, rather than to have an average age of 80, we will live until 160. Just because we slow down the process of inflammation. Now, if you take zonulin as a biomarker of inflammation, these folks, they found that, zonulin levels are much higher in, folks that they have, aging, no matter graciously or not, compared to young folks. And this goes vis-a-vis with inflammatory markers. So the more zonulin you have, the more inflammatory marker you have in the aging individual compared to young. And also markers of agility. The more zonulin you have, the less agile you are. So there is a correlation, again, between gut permeability, inflammation, and the process of aging. The one that I find extremely interesting is this study. So if you take ultracentenarian, and you compare to younger folks that had severe inflammatory process like precocious myocardial infarction, the ultracentenarian, they have much lower zonulin level than the much younger folks that underwent to an inflammatory process. Once again, suggesting that giving up the possibility to control antigen trafficking, therefore the acceleration of inflammation, is a key element of aging. So I'm going to finish the last few slides by saying, okay, and this is the question that everybody asks me, even today, what can I do to maintain my microbiome healthy? What can I do to really make sure that this relationship with microbiome is symbiotic and therefore I stay healthy? We're born once. We can take antibiotics 2-3 times a year. We eat 3-4 times a day. By far, food is the most impactful elements that control composition and function of the microbiome, by far. That's, again, something else that is not disputed anymore. They eat what we eat. And if you allow me a comparison that I typically give my students is, a clear sign of a healthy microbiome is a diversified microbiome. So in other words, that is well represented in all these components. The microbiome is like a farm with animals. We have cows, we have sheep, we have chicken and pigs and so on and so forth. They eat different things. We want to maintain a balanced farm, we gotta have a feeding habits to feed all these different animals. We have an imbalanced feeding habits. We put in disadvantage some of the folks, and we lose the chicken, and the farm doesn't make eggs anymore. That's the long and the short. Now, until in the early 1900s, the most frequent reason why we are sick and die were infections. This map, it is very worrisome. The main reason why we die and we got sick now is nutrition, not infection anymore, nutritionally related habits. This is the rate of mortality based on diet, the darker, the more, that we see worldwide, okay? And if you look at the composition of the problem, with the exception of the first one, and I can make even an argument, the first one. So high sodium diet, that is the most impactful, diet habits that will increase your morbidity and mortality, but that also will have an impact. All the others that you see here, diet loaded in whole grains, low in fruits, nuts, seeds, vegetables, blah, 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 affect the composition and function of the microbiome. And by the way, these are all socio-economic dependent. These are the diets that cannot be embraced by people that have no means. And by the way, this is the typical Mediterranean diet. Typical Mediterranean diet. That, by the way, once upon a time was called the diet of the poor. Not the diet of the rich, the diet of the poor. Why? When we were gatherer hunters, we eat a lot of fruits, a lot of vegetables, nuts, tubers. Why? They are there. Meat, oh yeah, but you gotta have a good hunter. You have to be a good hunter. And it's lean meat, these animals that escape predators, including humans. And this is a typical, one of the pyramids of the Mediterranean diet. By the way, it's not a diet, it's a lifestyle. Because that's another concept that I want to say. You can eat as good as you want, but you don't have a good lifestyle. That would be a waste of your energy. And in the Western Hemisphere, we don't grow old anymore together. We don't sit around the table again anymore. We don't take ourselves, too seriously. And we're all stressed out. And again, we don't exercise anymore. Those are all elements, together with a good diet, of a good lifestyle that will keep us healthy. I was born here, Salerno, right here. And the Mediterranean diet region is right there, the one that you see in green. I come from a very humble family. We were not wealthy. I didn't know that I was eating the Mediterranean diet. That's the only thing that we had available. We ate mainly fruits, vegetables, legumes, meat, yeah, but it was an exceptional event, that we ate meat maybe, I don't know, once every 2 weeks, because it's extremely expensive. Fish, yes, but the cheap one, so to speak, the blue fish, the ones that now we know that is so good for us. So that was our diet. And the folks that they are ultra-centenarian, not only they eat that way, they are not stressed. They walk everywhere. And that's the reason why we talk about lifestyle. And, to finish this up, have you guys seen Ratatouille, that movie? I love that movie. I'm going to skip to this because I don't want to waste your time anymore. So Remy and Emile are the two, examples of how you have the attitude towards food. Emil's feeling about food is fuel. You get picky about what you put in the tank, your engine is gonna die. Now shut up and eat your garbage. That's the Western lifestyle and attitude. Who cares? I can go to the freezer section of the supermarket, gut the trail, put in the microwave, and that's food. Well, you know, Remy's that is much more sophisticated. Feeling about food is if you are what you eat, then I want to eat the good stuff. And he said, anyone can cook, but only the fearless can be great. So, again, I think that this group is fearless because, again, you're doing this already. I think that, again, science will prove the basic of why we need to work toward a lifestyle that is focused on nutrition, because I truly believe that in the future we will see less drugs to fight inflammation and more nutritional habits. I'm going to finish by thanking, of course, you know, the folks that give us the money to do these studies, and a bunch of folks that, you know, I have, you know, dozens of people working in the lab. I'm just the orchestrator, but the real stuff comes from these people. And again, you guys for giving me the opportunity to chat with you today, okay? Thank you.
Thank you very much. Wow, that was fascinating. Hope everybody got as much out of it as I did. Thank you. That was really perfect, spot on exposure and survey of this field, and more to come. We're just still in the thick of it. I am getting some questions. We have a stack of them. Probably won't get through all of them, so let me go through as fast as I can. Thank you. Few questions on trials, clinical trials, because we are patients here and we do get exposed to opportunities to participate in trials or see what's in the pipeline so that we can maybe look forward to something that's coming that's transformational. So I have a couple of questions on that. What current studies are in process, and are there opportunities to participate in these studies? Who is looking at the SCD, specific carbohydrate diet itself, and its impact on gut microbiome? And on mental health as well?
Yeah, that's a very good question. The short answer is the best way to see what is cooking in terms of clinical trial is to go to this governmental, you know, site that is www.clinicaltrials.gov. If you go there and there are keywords that you can put there, like as, you know, a CD, and you will see anything that is going on there. So there is a lot of stuff that's going on in terms of nutrition and inflammation. You know, for example, one of the outcomes of these studies has been the use of what we call elemental diet as the first line of intervention for IBD, for Crohn's mainly. So as you know, we typically start with steroids to put the inflammation under control. And steroids have side effects. And this trial proved that, you know, elemental diet has the same efficacy than steroids to start, you know, controlling the inflammation, but no side effects. So now the new guidelines to start the treatment of Crohn's disease is an elemental diet. Now, of course, you know, there are a lot of stuff that's still being done, and, you know, people, they link this to the microbiome composition, so, yeah.
Yeah, clinicaltrials.gov is really good. Punch in zonulin, and you'll get a list of who's doing the study, who's the principal investigator, how to contact them, and then maybe look at the timing. They will then also publish interim results if they're available, or when they expect results. Now, one thing you mentioned was the specific carbohydrate and Mediterranean overlap quite a lot, right? But there's also disconnects, and one of the key disconnects is grains, whole grains, right? Wheat, whole grains. So obviously there's a lot of data supporting whole grains and whole grain diets in supporting health, Mediterranean diet style, microbiome, the fibers, and so on. That's an excluded component of SCD. So my question to you is, and somebody wrote this, is how do you fill that gap?
That's right.
And before you answer it, can I just also say that the data he showed specifically showed whole grain consumption
right.
is good. It means processed grain is bad, right? So in the context of that, you got to put, if you're eating processed white flour, you don't get the benefit. It has to be in a whole grain format, and then you get the benefit. But even SCDers don't do whole grains, so what do we do instead?
So out of curiosity, how many of you guys really have been physically in Italy on vacation? Okay. So if you've been there, the idea of mandolina, pizza, and pasta is not what Italy is all about. That's not the way that we eat. We don't eat a lot of grains as people believe they do. We do, of course, but it's part of a very balanced diet. If you do a Mediterranean lifestyle, again, now even in Italy, that is in the Western Hemisphere, has the poison of a Western lifestyle in terms of diet. But the real Mediterranean diet is not fettuccine Alfredo or spaghetti and meatballs. If you go there and you ask for that, they look like you come from Mars, if you go to a good restaurant, I mean. So again, no refined grains, but whole grains in moderate amount is part of the Mediterranean diet. So just to make the record straight. Yeah.
Thanks for that. It's an interesting story. And by the way, a lot of people eliminate gluten for no reason. Like they actually don't have celiac disease. So it's an implemented, not in the SCD community, but in general. That's why there's all these gluten-free foods. Okay, one question. So now, given the importance of the gut, colon microbiome, do you have any recommendations to help strengthen the microbiome of people with IBD who have had their colon removed, or other types of resection where there is no sort of host or home for these microbes? What happens then?
So the old exception was that the small intestine is sterile, the colon has the microbes. That's not true. We have a microbiome throughout the GI tract, but of course the vast majority, is in the colon. If you don't have your colon anymore, adjustment by Mother Nature are made, and part of the small intestine, the distal ileum, will become, ecologically speaking, very similar to the colon, particularly if the ileocecal valve has been part of what has been eliminated by, the colectomy, the colon that has been taken out. And not ideal, not identical, but very close surrogate ecosystem is created there. Maintaining a good ecosystem with or without your colon implies always the same kind of, principle that we discussed today. So you don't want a microbiome that is imbalanced because you lost some of the elements that can check, the others so that everybody lives in peace. So a good balanced diet in which you avoid refined stuff, junk food, and you favor fibers, containing food from fruits, vegetables, legumes, and so on and so forth, is the way that you please your microbiome.
So in essence, when you do go through that anatomical change, your microbiome adapts to the new reality, and then the game on again.
It needs to find a new niche, and it does.
Is the microbiome of a breastfeeding baby only as healthy as that of the lactating mom? Is there a known impact of breast milk if the mom is active IBS?
So again, inheritance, it's important. So if it's important that the genes that we inherited from our mother will dictate our life destiny, equally important is this parallel genome that is the microbiome that we inherited from our mother. We discussed this before. But again, coming to breastfeeding, now we're talking about refining what we receive from our mother in a specific way. So again, oligosaccharides are the same no matter what, and they will feed the good guys. But the breast milk also has its own microbiome that is passed, during lactation. And that can have an impact, for better or for worse, depending what is the situation in the mother. Now, IBS will require another 2 hours of discussion because now it's not even called IBS anymore. It's called gut-brain axis dysfunction because that's the cross-communication between these two organs that seems to be at heart about this. And, of course, the microbiome is part of the syndicate why you develop IBS, but not only that, there's much more at play there. Yeah.
We'll go through a few more here, if that's okay with you. Do you have a theory of why more women develop autoimmune disease than men?
So there are two non-mutually exclusive reasons. Genetics. So, men die earlier than women. So unless you want to trade in, keep your autoimmune diseases and let us die early of cardiovascular disease and tumors, because that's the difference there. Men, they have genes, they are less resilient to cellular repair, and therefore more susceptible to cancer and cardiovascular diseases. And women, they have genes, they are more prone to the autoimmune attack. And the reason why is because the autoimmune attack is due to the fact that the immune system, rather than fight the enemy, start to fight your own body. But that means that the immune system is very active in, surveilling, so to speak. It's just that they make a mistake in terms of target, and that's the reason why, women today develop less frequently other conditions that are typical of men. What was the second part of the question?
You got it.
Oh,
It's
okay.
just the incidence in women. It may also be the diagnostic.
The second element, that's right. The men, they don't go to the doctor as frequent as women. So if it's bearable, they stick it in wrongly, of course, autoimmune diseases. And that's the other reason why we die earlier, because we are now proactive in prevention and checking, what's wrong with us. So, yeah.
Two more questions. One is, what's the difference, sort of the underlying difference between true celiac, where you're actually immune reacting to gluten, and then to gluten intolerance without visible damage to the lining?
So clinically, they are not distinguishable because they give the same symptoms. But there's substantial difference in terms of the mechanism. So celiac and autoimmune disease, it's the immune system that goes after your intestine that's destroyed, and therefore all the digestive and absorptive machinery are jeopardized there, and you have clinical consequences. Non-celiac gluten sensitivity or whatever, with sensitivity or with intolerance and so on and so forth, is still an immune response, but it doesn't really imply an autoimmune connotation. So it is an inflammatory response that leads to the same symptoms, and is gluten dependent, but is not with the connotation of an autoimmune insult.
Back to zonulin. When do you think clinical utility of zonulin measurement, serum or stool, will be a reality? Are we there now? Is it overused? Is it misinterpreted? Is it coming?
So the problem is that the zonulin testing is done by some kits that they are still in a validation process, so to speak. Europe is now using routinely, most of the country in Europe, they use routinely zonulin as a biomarker of loss of barrier function and therefore permeability increase. And this is used routinely in clinical practice now, much more than we do here. Again, the problem is that, it's not a question if gut permeability is important for inflammation. That's also been agreed upon. The question is how we measure it, and we don't have really practical and stuff that you can do in clinic. And so far we use this double sugar test. It's very complicated, and Zonulin will offer simple tests to do that, mainly in the serum where it's more validated than the stools. But also, you have mentioned that it's a clinical trial. We have several ongoing clinical trials that we're doing right now, and measuring zonulin will give us a personalized target which of the folks will eventually respond to the trials. We're talking about trials with long COVID, rheumatoid arthritis, celiac disease, they are ongoing. And again, lastly, I didn't have a chance to mention this because I didn't have time. The same cohort of kids at risk for celiac disease that we've been following, they also, before the onset of the disease, they have an increased zonulin level months before. So what happened is that they have dysbiosis like 18 months before, 12 months before they have this increase in zonulin, then 12 months down the road they develop celiac disease. So this is now considered also a biomarker of prediction of disease, ongoing, that can be one of the biomarkers for preventive medicine in the future.
Thank you very much. I'm always willing to learn and excited to have you here. Thanks, everybody. Thank you.